This conference will now be recorded.
Okay, so I'm here with Baxter kind of leaning on me this morning. He's being needy today, so if you hear some weird panting in the background, that's just old Baxy. What we're going to do today is we're going to finish up the week. We're going to continue talking about special senses here. We're going to move on and kind of pick up where we left off last time. And where we left off last time, we decided that, or I decided, I guess, that we should probably hold off until now to get into this part because it's a little bit complicated. And the idea here is simply that the way that we see color has everything to do with the ability to separate the energy spectrum of light, okay, into different little sort of windows, I guess, and then to have a photoreceptor that is specific to those little windows. That's where we end up getting green and blue and red from. And if we look at kind of the entire spectrum of energy, right, as we understand it, right? So from very, very short wavelength things like gamma rays and X-rays through UV, and then to visible light, and infrared, microwave, and radio waves getting longer and longer wavelengths. What we're going to find is that, again, just the tiny little window of this total energy spectrum is what the human eye can see, right? And what we end up doing is having, again, these photopigments that are receptive only to a very specific region of this wavelength energy, right? So the shortest end of the visible spectrum, the narrowest wavelength here, is going to be where we get blue from. So we have one set of cones that's receptive to very, very narrow wavelengths. We have another set of cones that's receptive to these sort of intermediate wavelengths, and some that are way out here on the red end that are a little bit higher. And when light comes into your eye, right, the light that bounces off of whatever object that we're seeing gets refracted into different wavelengths, right? And depending on which of these wavelengths are present and how much, we can see things as red or green or blue or any color kind of in the mix between those two things. Okay. Again, it's very much like I don't know if you've seen some of those videos on the internet, for example, where we have people that, or screen printing for that matter too. I don't know if you're familiar with screen printing, but where we overlay color upon color upon color that are slightly transparent, we get shades of colors, oranges and yellows and blues and greens and purples and all of those things out of that. The other important thing here to kind of pull out of this, of course, is that we have only one photopigment for our rods, which again tells us that we don't get any color out of that. Everything is the same signal, and so we only see shades of gray, things that are lighter, things that are darker, but no color associated with that. Okay, so let's take a little closer look here at those photoreceptors. Again, rods and cones. Like the bipolar cells, like the ganglion cells, they're just modified neurons. Okay, what makes them modified here is that they've got this outer chunk called a receptive region where these proteins called photopigments reside. And this is actually what happens when we see light. Light comes in, it hits these photopigments, and it changes their shape, actually. And that change of a shape is what gives us the signal back to our brain that says we're seeing something. Okay, and we'll spend some time talking about that. So again, thinking about kind of the shape of the retina here, the inner layer here, right? So these are our bipolar cells coming down from the top, lights coming down in this direction here. These guys here, we have 3 rods in this picture, 1 cone. They're not necessarily yellow and purple, but they are shaped like this. Excuse me. And what's going to happen here, again, is as light travels down through here and it gets to this outer region and kind of the interface between the pigmented layer and the photoreceptor, is it will encounter these little photopigments in here, right? And deform them and change their shape and break them down. So because we are essentially bathing your photoreceptors in radiation, that is what light is, just like your skin, you know, if you spend too much time out in the sun, it's vulnerable to damage. Photoreceptors are vulnerable to damage too. Okay. In addition, you know, we talked last time about the idea of a detached retina, where a significant blow to the head, for example, might pull the retina away from the choroid, removing it from its blood supply. They will not survive without that blood supply, and so a detached retina can also be a source of damage to these things. Because they are so very important. If you hear that rattling noise in the background, that's our kitten Roger playing with a toy mouse. Because they are so vulnerable to damage and yet so very important, they are highly regenerative, to the point where the outer segment that contains the photopigments is essentially made brand new every day. Because again, they are being, you know, bathed by damaging radiation, but we need them to see. Specifically here, we'll start talking about rods, and we'll kind of move on to cones. Rods, if you remember, are extraordinarily sensitive, right? It takes very, very little energy, very, very little light to get them going. Okay, so for that reason, they're really, really good for, you know, night vision, right? Or not necessarily night vision, but, you know, dawn and dusk, periods of low light, right? Again, because they contain only a single pigment, we only get shades of gray, or that comes in gray scale, okay? And because they're kind of scattered across the retina, there's not a nice sharp picture. And they're all over the place. We don't get a nice focused bit of light in there, so everything's a little bit fuzzy. You know, again, the analogy that we use here is if you've ever seen some of those ridiculous ghost hunting shows or the Bigfoot shows or any of that kind of stuff, and they're out in the woods at night with the night vision cameras, that's kind of how your rods operate, right? Cones, on the other hand, right, again, remember, not particularly sensitive. They need very, very bright light, okay? So you get up in the middle of the night, you know, and the night light in the hallway is on. It's not very bright out. It's really only working your rods. It's not until you get into the bathroom, you turn the bathroom light on, and you go, oh, God, why did I do that? Right? That's when the cones actually become activated, okay? And again, we've got red and green and blue pigments in there, because we can have some or all of those activated at the same time. Now Roger has moved on to playing with the crinkly toy mat we bought him. Because those things are potentially overlaid, we get color vision from that. Also, because they're all highly localized, they're all in that macula lutea, where light is very much focused, we get very nice, sharp, crisp pictures when these cones are activated. Now, you may have heard of color blindness. This is typically a male-related trait. 99.9% or something of all colorblind people are male. Women can be colorblind. I've never met one, but they can be colorblind. And all this means is that one or more of those pigments simply isn't produced, or if it's produced, it's produced in a non-functional form. The way that these visual pigments work, right, these photopigments work, is actually really pretty simple. We take vitamin A and we turn it into a molecule called retinol. All right, and retinol combines with one of 4 other proteins. Okay, proteins called opsins. So there's a rhodopsin, for example, that's the rod pigment. There are red and green and blue cone pigments or cone opsins as well. And depending on which of those opsins we get, that's where we get our photopigments from. Okay. The way that this works is that retinol when exposed to light, changes shape. Okay, we call these different shapes of the same molecule isomers. And if you go on and you take any organic chemistry, you'll learn more about isomers than you ever wanted to know. For here, we're just going to kind of stick to the idea that one of these shapes is bent and one of these shapes is straight, right? The molecule itself has a kink in it, or it's all in one line, right? So the cis form is the bent form, okay? Cis here meaning kind of the typical or the standard form. And the trans form, right, is when it's transformed, right? When light hits it, it goes from being the cis bent form to the trans straight form, okay? And the bending of that molecule, or the unbending of that molecule, I guess, is actually what stimulates the sending of a signal back through the optic nerve to the brain. So we're going to use an example here just with rods. It works the exact same way with cones, okay, but we're just going to use the rods here as an example. Again, our pigment here, our opsin, anyway, here is rhodopsin, right? And so what we're going to see here happen with rhodopsin is a kind of a cycle, right? We make it, we use it, and we have to rebuild it. One of the things that people often don't know right off the bat is that you have a finite amount of photopigment in your retina, right? You can, in fact, use up all of it, all right? And if you use up all of it, that particular photoreceptor is no longer functional until you rebuild all of the photopigment in that, okay? And the conditions under which all these things happen are pretty simple. So this is the basic idea here. This is kind of a cycle of life. It's moved from the dark and into the light and back into the dark and back into the light. In the dark, right, in the absence of light, we can make photopigments. And I'm not super concerned with all of the chemistry here. The important thing here is that we're only going to get this cis form, or this bent form, right, in the dark. Okay, so we make it, this is called synthesis. We turn the lights on, right, you open your eyes, whatever, okay. We expose these cis photopigments to light, okay. They unbend, right, they go from cis to trans, okay, and this is called bleaching, right. And if exposed to significant light for significant periods of time, okay, we turn all of these forms, right, from cis to trans. Part of what happens here is we actually break the retinol away from the opsin. Again, the chemistry here is not super important. But the key here is that once we turn it from cis to trans, it's been used. And we can't use it again until we kind of rebuild it, right? And we can't rebuild it if there's light, because as long as there's light, it's going to prevent this form from being turned back into the cis form. So once we leave the light, once we go back into a dark environment, at that point, the final stage of our little cycle here, pigment regeneration, can take place. We can resupply ourselves with photopigments for the next time we're exposed to light. Again, with cones, basically the same process. Okay, really the only difference between how cones and rods work, again, comes down to intensity of light. It has to be very, very bright for cones to work, not so much with the rods. Okay, so here, when we talk about the eye being kind of maybe unnecessarily confusing or unnecessarily complicated as a structure, this is kind of where we're getting into that. This is kind of what we mean by that. What I'm saying here is that this three-person chain in our link can be a little confusing for people because we actually have a situation where we have to turn something off in order to see things. Confusing. Okay, so we need to revisit a concept from A&P1 here, the idea of EPSPs and IPSPs, right? An EPSP, right, is an excitatory postsynaptic potential. This is what we think of when we think of neurons talking to each other, right? One neuron releases neurotransmitter, and the next one on the chain gets stimulated, right? IPSPs, on the other hand, are situations where we inhibit postsynaptically, right? Where we stop the next neuron in the chain from releasing the neurotransmitter. The reason this is a little bit wacky, okay, is that neurotransmitters, right, photoreceptors here that are the photoreceptors in your eye, when they are inactive, okay, so when it's in the dark, they are constantly releasing neurotransmitter, okay, and this neurotransmitter is inhibiting the bipolar cell, right? So photoreceptors in the dark are generating IPSPs. They're stopping the bipolar cell from talking. All right. When we expose them to light, we go from cis to trans and we break the opsin off of the retinol. Okay. This causes the photoreceptor to hyperpolarize, to stop releasing neurotransmitter. Okay. And because we stop releasing neurotransmitter, we allow the bipolar cell to talk to the ganglion cell and send a signal back to the brain. So here's our 3-neuron chain, right? Photoreceptor up here, bipolar cell in the middle, ganglion cell going back to the brain down here. Okay. In the dark, photoreceptor constantly talking, right? Constantly inhibiting the bipolar cell, and the bipolar cell cannot release neurotransmitter, so no signal gets sent along the pathway. In the light, right, we inhibit, we hyperpolarize the photoreceptor, we stop releasing neurotransmitter here, we allow the bipolar cell to release its neurotransmitter, to talk to the ganglion cell, to send the signal back through the optic nerve to the brain. So it's, again, very, very unusual as far as our sensory processes go. This is kind of an odd thing to do. So sometimes it gets a little confusing, but that's the idea. The idea here of light adaptation is something you're familiar with, even if you aren't kind of aware of the terminology here necessarily. Light adaptation and its sort of opposite coin here, dark adaptation, are basically just the processes that go through as you transition from one state dark into another light, or vice versa. Okay, so for light adaptation, we're moving from darkness into bright light. When we move into bright light, we go from a state where none of your photoreceptors, or very few of them, are active, to one where all of them are active. All of a sudden, all of your rods, all of your cones, bam. Okay, the immediate response for your eye is to constrict your pupil, or to go, oh shit, this is too much light, we've got to slow this down. So they constrict the pupils way down, but it's still a little bit too late, right? We've still already gotten in and had this massive flare of photoreceptor activity, right? This is why when you walk from a dark room into a bright room, your eyes hurt a little bit. You can't really see anything. Everything kind of seems washed out and bright, and you can't really see. Okay, this is because your brain is being overwhelmed, right? All of this signal at once, and your brain just panics. It's like, oh my God, what am I supposed to do with this? I can't do anything with this. And it really only gets better, and it takes maybe 5 or 10 minutes sometimes. Really only gets better once you've actually bleached all of the rod pigment. Okay, once you've gotten all of those rods basically burned out, and there's no more photopigment, or very little photopigment left for them to kind of burn through, now you're getting less signal sent back to your brain again. Now your brain can work with it, can parse it, and you get back into a position where you can see things again, right? The opposite side of this, dark adaptation, is when you go from a bright light into a dark room. Okay, now you've got a problem. Okay, there's not enough light for your cones to work, so they turn off. And you were just in a bright room and you've bleached all of the photopigment out of your rods, so they can't work. So you are at this moment in time up shit creek, basically. You can't see anything. Okay, and it's going to take you 20 or 30 minutes, okay, in this dark environment to regenerate, right? The third step in our little cycle, to rebuild all of that rhodopsin so you can see again, right? So it takes a significantly longer time to rebuild photopigment than it does to bleach it all out, right? 5 to 10 minutes to bleach it all out, 20 to 30 minutes to rebuild it to the point where you can see again. So this takes an awful lot longer to get used to. And again, these are things that you're at least subconsciously aware of, even if you don't know the processes behind them. All right. Here again, just to kind of clean out maybe the last of the potentially confusing things, we talked before about the idea that vision is contralateral, right? In the same way that all sensations are contralateral, right? Things come from the, I touched the palm of your right hand, that signal goes to the left side of your brain, right? And vice versa, of course. With vision, the idea is similar, but not quite exactly the same thing. Okay, and this is for actually a really very important reason. When we talk about vision, what happens is the information from your right eye doesn't go to your left brain, and your left eye doesn't go to your right side. What we're going to do is we're going to take your field of vision, all right? So everything that you see with your right eye and everything you see with your left eye. And what we're going to do is we're going to take that field of vision, okay, and we're going to divide it in half into a right side and a left side. And so everything that you see on the right side of the room from your right eye, okay, goes to the left side of your brain. And everything you see on the right side of the room from your left eye goes to the right side of your brain, or left side of your brain, excuse me. So if we look at a little diagram here, right, here's our field of vision, right, and we've color-coded this right side in blue, left side in yellow. And we see that both eyes see parts of both sides of the room, right? The left eye here sees a lot of the left side and a little bit of the right, right? And the right eye sees the left side and a little bit of the right, okay? What happens is all of the blue, right, all of the right side of the room goes to the left side of the brain. And all of the left side of the room, here in yellow, goes to the right side of the brain. The reason this is so important is that this is what gives us three-dimensional vision. Well, it gives us depth perception. Okay, and the way that your brain figures out how close or how far away something is it compares the signal it gets of the same object, right, from the right eye and the left eye. And you can do this yourself. If you sit here and you look at an object about 3 feet away, right, close your right eye, right, and then close your left eye and open your right eye back up, right? You see a different angle, right, of the same object. And if you look at something really close, right, and then something really far away, what you find is that the closer the object is, the more different, right, the angle that you see from the right eye and the left eye is, right? And so what your brain does is it says, okay, I'm going to compare the same object. If it's really different, I know it's close. If it's basically the same, I know it's far away. And it can't do that, right, unless the same part of the brain gets both of those signals, right, from the right eye and the left eye. So that's why we divide it into field of vision rather than from one eye or the other. Right. And again, this is kind of the basic idea there, right? Because we see everything from a slightly different angle from each eye, right or left, depending on how far away it is, this is where we get that depth perception from. Not super worried about the visual processing stuff. I think we're just going to kind of move past this. What I do want to talk about here, though, is what happens in the brain in terms of identifying various aspects of what we're seeing. Okay, so if you remember from ANP1, the primary visual center, right, is in the occipital lobe, way at the back of the skull, or back of the brain rather. This is where we begin to process shape and color and movement. But more complicated questions like what and where, right, occur in other parts. So in the temporal lobe, right, in the ventral part of the temporal lobe, this is where we actually figure out what something is, right? So if I'm looking over here at our kitten Roger, who is fighting with our other cat Penny now, for other reason, they're kind of play fighting. Identifying that as a cat, identifying that specifically as Roger, that's ventral temporal lobe. Seeing that they're over there in the dining room, okay, just right next door to us here, 6-8 feet away, that's going to happen actually in the parietal lobe, okay. Between the parietal lobe and the postcentral gyrus there is going to give me spatial location, how far away, all that kind of stuff, what direction from me, all of that. And both of those things happen to then communicate with the frontal cortex, where we decide what we're going to do about it. Okay. We're going to then move on to two related special senses. We call them collectively the chemical senses here, for reasons that will become obvious. Smell and taste. Smell and taste operate more or less the exact same way. They're perceived in the brain basically right next door to each other. Okay, and the location of the senses in your head are very, very close to one another also, right? Your nose and your mouth. And they're linked for all of these different reasons. Smell here, technically we call this olfaction. Taste is gustation, okay? And the reason we call them chemical senses is that in order for either of them to work, we have to have a small particle of whatever it is, okay? A smell or a taste, dissolved in a watery or an aqueous solution, okay, saliva or mucus, and actually bind to a receptor protein on the receptor cell in order to actually smell or taste anything. We're going to start with smell, with olfaction. No particular reason, we're just going to start here. And with the sense of smell, obviously this is going to occur in the nose here, specifically in the olfactory epithelium. I'm just going to show you the pictures here, and we'll go back to the other pictures for a second. The interesting thing about the olfactory epithelium and about how the actual sense of smell occurs is that we don't usually see situations where the sensory structure itself, or the sensory neuron, the receptor, is exposed, right? Is sticking out of the epithelium, right? Invariably, aside from this case, they're embedded within tissue, right? Some of them are encapsulated, some of them are not, but they're embedded within tissue. They're tucked away, they're hidden away. Here with olfaction, this is not the case, right? And if we look at this particular little cartoon here, we see here these big, tall cells here, right? These columnar cells of the olfactory epithelium. And we see that the sensory neurons themselves, they actually stick out. They're directly exposed, right? They hang out. And the only thing that protects them, right, is this little thin little layer of mucus on the surface of the olfactory epithelium. Okay. And they, of course, they extend. The cell bodies, of course, exist here in the epithelium, but the axons continue and actually pass through the ethmoid, right? We talked about the cribriform plate, the ethmoid before. They actually pass all the way up through those holes in it, right? And into the olfactory bulb here at the end of the olfactory nerve, of cranial nerve number 1. So unlike vision, we have a very simple little relay here. Again, so there's another small difference there. And if we look at kind of what ends up happening here, right, we go directly from there to the olfactory bulb, the olfactory tract, and back to the brain. Now, you and me, we can distinguish somewhere between 10,000 or so different smells, right? Baxter here, next to me, and our third cat, Nola, we have a lot of cats, kind of giving me the side eye as I say that right now, because they, of course, can individually smell significantly more than 10,000. We're pretty lousy smellers. But in our defense, we distinguish 10,000 different smells with only 400 different smell proteins, right? With only 400 different receptor proteins, we get 10,000 different smells out of it. And the way that we do that is, again, kind of like we talked about with vision. We mix and we match, right? The same odorant, right, which is the chemical that we use to smell, can bind to more than one different receptor. So, and at the same time, every protein can bind to more than one different odor. So by kind of mixing and matching our signals, we get distinct smells out of that. If you go outside today, for example, when it's, I think, 12 degrees here on the mountain, you will also be intimately aware that you have pain receptors, right? You have nociceptors, you have temperature receptors, also in your nasal cavities. They're not so much in the olfactory epithelium, but in the remainder of the epithelium within there. Again, the physiology here is pretty simple, right? We have to have a particle, right, an odorant, okay, in the air that we breathe in. It has to encounter the mucus. It has to dissolve in the mucus. And once it's dissolved in the mucus, it has to encounter the correct receptor protein on a olfactory neuron. And so this is kind of why we have this unusual situation where the olfactory neurons are exposed. As you might imagine, unless you go back in time to Pittsburgh in 1980, the air that you breathe in doesn't have a lot of stuff in it, right? You can't chew it, right? The smells and things that you get are extraordinarily rare particles in that air. And so we can't have a situation where it's hard to encounter an odorant. There's just not that many of them, right? And so we have our receptors exposed, right, to maximize the opportunity to actually encounter them, right, to maximize our chance of smelling something. So that's why we have those things exposed. I'm not too worried about most of this stuff. I do just want to talk to you just real briefly about this last concept here, olfactory adaptation. We've talked about adaptation. We talked about general senses, the end of ANP1. Here, the idea is basically the same thing, right? You know, you go to grandma's house, she's making cookies, you're like, oh shit, this smells awesome. And then eventually you can't smell them anymore, right? It's not because grandma stopped smelling cookies or stopped baking cookies. It's not because the cookies stopped smelling. You simply just adapted to it. Your brain is like, yo, I get it. There's cookies. Let's move on, right? You don't got to tell me about cookies all the time. If we didn't do this, it would be like living with a 3-year-old, right? I don't know if you know any 3-year-olds. They tend to tell you the same thing again and again and again and again and again. When my son was 3, I probably heard about the Paw Patrol guy 100 times a day, right? I love my kid. I don't need to know about the guy from Paw Patrol that much, right? Your brain is the same way. It doesn't need to know that there are cookies as long as there's cookies. You heard about it once, you didn't forget. And so we have this capacity to do that. Now, not every smell actually becomes adapted. So there are smells like sulfur smells, like skunk odor, for example. If you've ever had a dog or a cat that attacked a skunk, you are well aware that skunk smell does not adapt. Okay, and this is the reason that we put sulfur smells in natural gas, so that if you are asleep and there's a gas leak in your house, you wake up and you smell the gas, right? If you adapted to it, if the gas leak happened while you were asleep, you might not smell it anymore. You might have adapted to that smell when you got up, and then you would be basically dead. So there are some smells that you do not adapt to, but most smells you do. Oh, not worried about this, I guess. Um, Last thing we'll talk about today, and we'll move on to the weekend here. Taste buds. Taste is a chemical sense, right? So it works essentially the same way as olfaction does. Um, Only here we have our receptors, and there are about 10,000 different taste buds in your mouth. A little more if you include all the ones that are kind of around the mouth. Um, but these taste buds. These are where we have taste receptors, and these are actually, unlike olfactory receptors, these are hidden away. They're tucked away. We'll see kind of a picture of these in a little bit. They're actually kind of hard to get to, right? And the reason for this, the reason they're hard to get to, is because when you put a piece of steak in your mouth, it ain't rare. Well, I mean, if you have steak and it's rare, of course, that's the best way to have a steak. Um, but the tastants, right? The things that make the steak taste like steak, they're not hard to find. And if your taste buds were exposed the same way that your olfactory receptors are exposed, you would be overwhelmed by taste. It would be like when you walk into a bright room and your eyes are like hurting and you can't see anything. That would be the same kind of a scenario, right? So we tuck these away, we hide them away in taste buds so that we can still taste things, but we're not overwhelmed by them. Most of these taste buds are on your tongue, okay? We do have some on the roof of your mouth and soft palate. We do have some on your cheeks. We do have some all the way back on the pharynx, the oral pharynx. Um, we do have some on the epiglottis, okay? But for the most part, they're on the tongue, right? And if we look at the tongue here and parts of the pharynx and the epiglottis, for example, we see they come in a bunch of different forms, right? We have fungiform, which are the ones on the tongue surface itself, right, that you see. We have foliate ones on the side of the tongue in the back here. We have these valate ones here in the back, these large ones back there. But again, scattered kind of all across the tongue. One sort of old wives' tale that you might have learned in elementary school, primary school, at any point, I guess, is that there are different regions of the tongue that are specialized for different tastes. That's actually not true. Basically, every part of your tongue is capable of tasting all of the different tastes that you can encounter. Here we're just going to kind of zoom in on one of these valate papillae back here to show you what I mean by kind of having them tucked away. The taste buds themselves are actually buried deep within the tongue epithelium here, and are only accessed through these little crevices, these little, you can see maybe a better view here. So that for a taste to occur, some little particle of it has to work its way down into this crack and then encounter the taste bud from there. So again, they are tucked away. In terms of how these things are built, I'm not overly worried about this slide. In terms of how they're built basically out of two different kinds of cells. We have taste cells here, the gustatory cells, and we also have these basal cells in here as well. Again, just providing structure. Also going to be there to generate new taste cells. We do rebuild these taste cells, every week or so, right? Just because they do eventually wear out. The other thing here to point out about the taste bud is that not only do we have to work our way down into this crevice here, but once we're inside that crevice, in order to actually access the taste bud, we have to pass first through this tiny little hole here called a taste pore. So again, it's not an easy thing to do to actually get to a taste bud. Again, simply because we have this sort of embarrassment of riches in terms of tastants in the mouth. It's very much unlike the scenario with the nose, where odorants are very rare. Here, they're very, very common, too common, and so we have to kind of restrict access to these taste buds. Now, one thing you've probably learned is that there are 5 tastes: sweet, sour, salty, bitter, and umami, which is a sort of a meaty taste. Sweet, we associate this with sugar, obviously, with saccharin, sort of fake sugar. Alcohol, right? Ethanol, anyway. Some amino acids, lead salts, which is why kids used to eat lead paint, because it was sweet. Sour, we associate with acids, right? So the release of hydrogen ions, right? So acid, right? So things like, citric acid, stuff like that, things are sour. Salty tends to be metal ions. Sodium is a good example of that, but potassium or any of those sorts of inorganic salts. Bitter things tend to be things that are poisonous. So plants produce alkaloids, alkaline basic. So rather than acids like sour, bitter is basic. That tend to be poisons. So nicotine, quinine, aspirin, even though we use some of these things as medications, they are produced by plants largely as poisons. And then umami, because it's a meaty taste, right? This is going to be amino acids. This is going to be proteins, right? So particularly here, glutamate and aspartate. This slide's a little bit out of date. It's really kind of no longer debated. There is a 6th taste. This is fatty. This is why fatty foods taste so bloody good. You can actually also respond to long-chain fatty acids, right? So we also have fatty here as well. And there's a different term for this that they've used. I just call it fatty. It's fine. And along sort of the picture here of tastes, liking different things or having craving for different things have some intrinsic homeostatic value. We talk about alkaloids largely being plant poisons. Things that are bitter are not pleasant tastes, right? And so they're there to kind of prevent you from eating things that will kill you, right? Along the same lines, your body needs a significant input of carbohydrates, right? Things that are going to come out and taste sweet. And so things that are sweet taste good, right? Or things that are proteiny, right? That have an umami taste good, right? And so it's kind of your body's way of saying, eat these things that won't kill you, that we need. Don't eat these things that will kill you, that we don't need. There is also some evidence to suggest that cravings, right? So we talk about pregnant women cravings, and all the hilarious cravings, right? All that kind of sitcom-y shit. But there is some evidence to suggest that if you have a craving for something, it is indicative of a deficiency in your body. This can be expressed kind of most extremely in cases of pregnant women eating dirt or eating rocks or pebbles or sucking on pebbles because they have mineral deficiencies. This is really, really, really common, particularly in Eastern Europe, where nutrition is not necessarily always highly available. And so you'll see women eating clay and things like that. Sometimes you also see small children doing these things. Sometimes it's just because small kids do dumb shit. Sometimes it's because they have a mineral deficiency and their body is trying to encourage them to rebuild that. Okay. All right. So we'll wrap this up here. We'll come back on Monday for half of you anyway. It will be face-to-face. The other half of you, if you go to the streaming link on the Canvas website, you will be able to stream this video live. Of course, if you're in section 5, we're all face-to-face, so there's no streaming required. All right, have a good weekend, and I will see you guys on Monday.